So for these next set of slides, it's going to be the process of how the nervous system and the cells we just talked about and the duodenum are going to act on food as it enters the stomach, as it leaves the stomach. Now, for the first time ever, people with red-green color blindness win. Because, yes, because for today only, red and green arrows mean the exact same thing. There's no difference in the meaning between red and green arrows. I don't know why these slides did that. I mean, I do. It's step 1 versus step 2. Step 1 is red, step 2 is green, but that's of almost no significance to you whatsoever in understanding this. So both of these arrows are stimulatory. They are both excitatory. They are increasing the action of the thing they are pointing to. Yay. The only thing that is inhibitory, the only thing that decreases the action of the thing it's pointing to is this symbol right here. It happens to be in blue, but it's got this little kind of stop icon at the end of it. So far so good for what those things mean. That's like the key to understanding these slides. So again, don't overthink it. It's just steps 1, 2, 3. Step 1 is cephalic, step 2 is gastric, step 3 is intestinal. Before you even have food, you start thinking about food. And that thinking is how we get the name cephalic. The cephalic phase means you start thinking about food. And that is going to cause your central nervous system to send a signal along our favorite parasympathetic nerve, which would be what? Vagus! And that vagus is going to innervate the submucosal plexus of the stomach. And the submucosal plexus of the stomach is going to stimulate mucus cells to produce mucus, chief cells to produce pepsinogen, and parietal cells to release hydrochloric acid, as well as those G cells, those gastroenteric cells, to release gastrin. So that's step 1. Even before you've started eating, your stomach is starting to secrete the products that are going to help you break food down. In the gastric phase, now food has entered the stomach. The food bolus has entered the stomach here. There's going to be several things that are going to continue the actions of these cells that we just talked about in the last slide. For one thing, as food makes it into the stomach distends, it stretches, and those stretch receptors are going to provide feedback to that same submucosal and myenteric plexus of the stomach, as well as chemoreceptors detecting the elevated pH from the hydrochloric acid. So those are going to be in a feed-forward or positive feedback loop with the submucosal plexus to further increase the production of mucus, pepsinogen, and hydrochloric acid. That gastrin that was released in the last slide is also going to increase the activity of chief and parietal cells, as well as increasing those mixing waves. So the rhythmic contraction of smooth muscle in the stomach is increased by gastrin. In stage 3, we're saying that food is exiting the stomach into the duodenum. The duodenum is the first part of the small intestine. So the duodenum detects the presence of nutrients, lipids, and carbohydrates, and a change to pH. And the duodenum releases several substances, 2 hormones at least, hormones, chemical signals, CCK, and you'll see these spelled out on other slides. This is cholecystokinin, GIP, gastric inhibitory peptide, and secretin. And those are going to inhibit chief and parietal cells as well as peristalsis of the stomach. At the same time, stretch and chemoreceptors of the duodenum are going to inhibit the myenteric plexus. And that's it. On your worksheet, what you're going to focus on is how these things act in response to stomach activities exclusively. So you will see that CCK, GIP, and secretin improve absorption and trigger some things that are excitatory within the small intestine, but they are inhibitory towards stomach activities, and that's what you're focusing on. How are these things acting on the stomach specifically? Because once things stop in the stomach, they have to start in the small intestine. Does that make sense? Okay. So the small intestine finishes the chemical digestion process. There is a difference between chemical and mechanical digestion. Mechanical digestion is the stomach going hump, 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 whereas chemical digestion is hydrochloric acid, pepsinogen, lipase, amylase, all of those enzymes that help break things down. That's chemical digestion. So the stomach is not about absorption of nutrients. It's about digestion and breakdown of nutrients. The small intestine is actually going to be about absorption. We're actually going to absorb our nutrients through the small intestine. So in a living person, the small intestine is actually pretty short due to muscle tone. It's only after somebody is deceased and they lose their muscle tone that 20-foot long small intestine is actually a thing. And of course, ingested materials, spending 12 hours as an estimate. It's highly variable between individuals. Yeah.
So in a volvulus, that would be in the small intestine.
I think volvulus can actually occur in small or large intestine. I think it's more common in small intestine, but I think that ileocecal junction is actually a common place for it. Yeah.
So if they're taking out the intestine, they're taking out that whole thing, probably.
In terms of volvulus or in terms of removing portions of small intestine for disorder? What are we going at for?
Well, I'm thinking of my daughter who has the malrotated intestine. When they said they took out the intestines, they're taking out all of it probably, and then they had to reorganize it.
Oh, to remove it physically and then reorganize it and then put it back, possibly. Yeah, I've heard of that for other surgical procedures, removing just florp everything, but yeah. Doesn't seem like the most effective way to do things when it's literally just a twist. Yeah. Okay. Regions of your small intestine. You can't just say small intestine anymore. You have to be more specific. You have a duodenum, a jejunum, and an ileum, in that order. And have a little song. You guys want to hear a little song? Duodenum, do 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 do, jejunum, do 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 do, the ileum, do 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 do, etc. You get it. Okay. The duodenum is retroperitoneal. It's closely associated with that head of the pancreas. We're going to learn in just a bit that pancreas is going to secrete some digestive secretions into that duodenum. They've actually got a duct directly into that duodenum. So since that pancreas is retroperitoneal, the duodenum is also retroperitoneal. It's actually got a little flexure right here. And in humans, this is something you can feel. It comes out at an angle. And that flexure is something palpable. So it's a very clear division between the duodenum and the jejunum. That jejunum is intraperitoneal. So we've actually gone through the peritoneal membrane at that duodenojejunal flexure. And now we're in the jejunum, and the way this is being portrayed is the jejunum is largely upper left, whereas the ileum is largely lower right. In real life, it's not going to be that clear cut. There is a histological difference between the jejunum and ileum, which is why we have different names for them. I mentioned earlier plicae circularis, or circular folds of the small intestine. They are much foldier in the jejunum. The ileum, by contrast, is much smoother. So we actually go from an area of much more capacity for absorption in the jejunum, and then we gradually transition to an area that's less folded and less capable of quite that amount of absorption. There's still absorption taking place. It's just we're trying to get as much of it early as we can. So that's the real difference between jejunum and ileum, and it is a gradual transition in real life. But, you know, you've got a nice color-coded upper left jejunum, lower right ileum for testing purposes. Back in the day when I had lab practicals where you would actually go in and have to identify it on a person, I would always tag the ileum right over here, close to the ileocecal junction. So this is the cecum over here. It's part of the large intestine. And you have that transition from small intestine to large intestine right here at the ileocecal sphincter. Notice it says valve right here. This is a pet peeve of mine. Does that look like a valve to anyone? No, it's a sphincter. It's circumferential muscle that contracts to close that space. That actually is a problem that I have with a lot of things. Actually, let's go back for a second. I'm sorry I didn't talk about these sphincters before. This is the cardiac sphincter, and this is the pyloric sphincter. And this is nice enough to call this a pyloric sphincter right here. Cardiac sphincter up here, or lower esophageal sphincter. I've been using sphincter all day. Sometimes people do call these pyloric valves and cardiac valves or lower esophageal valves. There's nothing valvular about them. So it's just a pet peeve of mine. If you say valve, I technically can't say that you're wrong about it because other sources will call it valves. It's just a sphincter. I know nobody likes the word sphincter, but it's just not a valve. Just don't do it, please. Okay, so ileocecal sphincter, not valve, is right here. So if you tag that ileum right here near the ileocecal junction, you know for sure that you're an ileum because the jejunum simply does not attach to the large intestine. That's all. That's how I would have done it back in the day before a nice color coding like this. And if you don't have the benefit of seeing these colors as different, then that's the other way to do it, is the ileum is close to the ileocecal junction. It's in the name ileocecal junction, so it makes it easy. All right, so we made it from the duodenum to the jejunum. We absorbed a whole bunch of nutrients here. We made it to the ileum. We made it through the ileocecal sphincter. There's a little dead end here known as the cecum, and then it's going to go from the ascending colon, transverse colon, descending colon, sigmoid colon, rectum, and then anus is the exit. So histology of the small intestine. Circular folds, or plicae circularis, are the macroscopic folds. You can see them. Those are covered in villi, and the cells of those villi are covered in microvilli. So again, you can see the circular folds, or plicae circularis. They'll look a little bit bumpy, so to an extent you can see the villi. But histologically, those villi have cells that also have microvilli. And all of that serves to increase surface area, increase our ability to absorb proteins, fats, carbohydrates, liquids, and then in the large intestine, vitamins and minerals as well. What's the difference between microvilli and cilia? Cilia move. Do microvilli move? Nope, microvilli are stationary. They are just about surface area. Okay, we made it to the large intestine. Now it's feces. It's no longer a food bolus. Chyme is what it was. It's not quite chyme. I guess we call it chyme after the stomach. Let's see. Bacterial flora are throughout the GI tract, but they are housed mainly in the large intestine. There are some little details I'm going to point out here. That bacterial flora is going to help you synthesize B vitamins and vitamin K in your large intestine. When you think of your large intestine, I do want you to think of vitamin K and B vitamins. It also is going to help ferment indigestible carbohydrates. So if you're a vegetarian, if you have a primarily vegetable diet, it turns out humans don't actually have the enzymes to benefit from all of the nutrients in vegetables. But some of our resident bacteria might be able to. So in order to get nutrition out of vegetables and actually benefit from vegetables, we have to ferment them with our bacteria. And that happens mainly in the cecum. And the human cecum is actually relatively short compared to many other animals. Many animals that are omnivorous or herbivorous have much longer cecums and thereby have much more ability to ferment vegetable matter and much more ability to get nutrients out of them than humans do. So if you have considered vegetarianism, maybe you've looked into the history of what humans are supposed to eat evolutionarily. It's kind of supposed to be a mix between vegetables and meats and other things like that, right? There's arguments for both sides. So we've got these grinding teeth, we've got these grinding molars, we're supposed to be herbivores. We've got canines, therefore we're supposed to eat meat. It really is a mix of those two things. The short cecum is a strong argument for, no, we're not supposed to live exclusively on vegetables. That is absolutely what that means. Okay, what else do we have? Some more anatomy of the large intestine. Again, I mentioned the cecum. That appendix is a dead end even further off of that dead end of the cecum. So there's the appendix. Ascending colon, we call this the right colic flexure. We call this the left colic flexure. There's actually, I think we also call this the hepatic flexure versus the splenic flexure, if I recall correctly. Transverse colon, descending colon, sigmoid colon, rectum, and anus out here. And then, thank goodness, your anus has an internal smooth muscle-based sphincter, but also, this is the part I'm grateful for, an external skeletal muscle sphincter. So this one's under autonomic control. This is when you feel like you need to go use the toilet for fecal purposes. Whereas this one's skeletal muscle, that can hold it closed even after it's time to go. So we have some bowel control thanks to that skeletal muscle. Thank goodness for that. Again, this is autonomic innervation. The internal anal sphincter, the external anal sphincter is under voluntary skeletal control. More anatomy here. These individual segments of the large intestine are known as haustra. You also have some little fatty sort of drop-shaped things known as omental appendages or appendices. So people just have a sort of variable amount of fat hanging off of this strip right here. What's this strip called again? I don't think it's on here. Tinea coli is what that thing's called. I think that's about it. Large intestine, you've got a lot of goblet cells secreting mucus. You still have the same layers. You still have mucosa, submucosa muscularis, and largely adventitious. Or sorry, largely seroses. Valsalva's maneuver, I could do some really nifty hand motions here if we wanted to. It's going to aid in defecation. You close your glottis over your lungs, and that's going to help increase the pressure in the thoracic cavity, which is going to put downward pressure on the abdominal cavity, along with contraction of abdominal wall muscles. That increases your intra-abdominal pressure on your large intestine. Then you use your levator ani muscles to actually lift the anal canal so that feces actually drop out as a result of that lifting motion, which I know those hand motions are fantastic in conjunction with this topic. Okay, questions about large intestine, defecation? Again, purpose of large intestine is related to vitamins B and K, absorbing water, dehydrating that chyme into feces is the purpose of the large intestine. Moving on to liver. Your liver is actually divided into 4 lobes. There's 2 major ones, the right and the left, and there's 2 smaller ones on the postero-inferior surface. And that is the quadrate lobe, it's very square-shaped, and the caudate lobe, probably because of its caudal position. Do you guys remember what caudal meant? Rostral caudal? Towards the tail, perfect. It's worth noting that we are closely associated with the inferior vena cava when we talk about the liver. We've talked about hilums already, right? So you have a hepatic hilum as well, and it's going to be the entry point for ducts, arteries, and veins. Hepatic portal vein has an entry to the liver here at this porta hepatis. Also note the location of the round ligament of the liver. This is separate from round ligament pain. If you've been pregnant, you also have a round ligament of the uterus, so that it's not the same ligament. They all just both happen to be round in shape. Anterior and superior is the falciform ligament of the liver. Note that there's this duct here coming from the liver, and then you have a cystic duct on the way to the gallbladder, and then you're going to have a common hepatic duct that leads down towards that pancreas. The liver does so much that anytime I have to come up with an exam question along the lines of what does the liver not do, it's been a real challenge for me, and what the strategy I've ended up having to have is what is a task that is very specific to a different system. So, I mean, it just does everything. It's going to actually be the one that synthesizes bile. You may have heard that bile comes from the gallbladder. It does. The gallbladder actually concentrates and stores the bile, but it's synthesized by the liver. The liver is actually making it. The gallbladder is just holding on to it for a bit. It's going to store glycogen and lipid reserves. It's going to be really very important to blood sugar control. So it's going to control how much blood sugar you have at any given moment, along with your pancreas. It's going to be communicating with your pancreas to control your blood sugar and nutrient levels, because it's responsible for a lot of your metabolic activities. It's going to take in all of the nutrients from your digestive system and filter them and alter them, metabolize them, store some of them, release others. It can interconvert nutrient types. Maybe you're hearing some of these trendy diets like ketogenic diet or paleo diets that are really high protein, low carb. And the idea with a lot of those is if we stop eating carbohydrates, our body has to take that nutrition from fat, and then we'll lose weight, supposedly, right? That's called interconversion of nutrients. That means we can take fat and put it into that cellular respiration cycle and still get energy out of it, even though we didn't consume glucose, which is what we normally put into cellular respiration. Does that make sense? Does that tie in a thing? Okay. Cholesterol levels, inactivation of toxins, including alcohol, including your drugs. It's going to be called the first pass effect of your liver. Storing iron, storing fat-soluble vitamins. Some vitamins are water-soluble. You pee out the excess. Some are fat-soluble. You store excess in your liver. It's also going to synthesize plasma proteins. You guys remember the plasma proteins? What were they? Albumin. It's good that you remember albumin. Gold star for remembering albumin. That's the important one. I mean, they're all important, but that's the first important one. That's the dead horse one. Yep, fibrinogen is the one for the clotting. Probably our second most important for this context. So you're doing good. 2 out of 3 ain't bad. Anybody remember number 3? It was globulin. And that includes immunoglobulins, which are antibodies. Okay, plasma protein synthesis. Synthesis of clotting factors. So in addition to fibrinogen, it's also going to synthesize, I think we pointed out factor 8, 9, 10 on that clotting cascade when we did our blood lecture. So you know clotting factors are actual individual proteins that are necessary for the clotting cascade. It's part of our RAS system. Angiotensinogen is secreted by the liver. Phagocytosis of damaged red blood cells. What other organ phagocytizes damaged red blood cells? The spleen. So it's going to act like a second spleen, and same with blood storage. The spleen acts as an organ of blood storage, so does the liver. So even if you don't have a spleen, you can still do some of those things. It might be a little bit stressful for the liver to take that over exclusively. Absorption and breakdown of circulating hormones like insulin and epinephrine. If you are in fight or flight this morning, but you're no longer running away from a bear, do you want to still have epinephrine coursing through your veins? No, we got to break that down eventually, and the liver is going to do that for you. And absorption and inactivation of lipid-soluble drugs. And again, we already talked about inactivation of toxins, drugs as well. So it's just a huge list. Yeah. Ligament venosum, porta hepatis, is the hilum, so to speak, of the liver. Histologically, your liver's really cool. It's actually in a hexagons, little stacked hexagons. This actually, the way I drew this was not a mistake. I drew a little hexagon because these are blood vessels in the liver. So this is a branch of the hepatic portal vein. Let's look at that hepatic portal vein really quick. Do you guys remember what a portal system is? We've only seen one other before. When we saw that other portal system, I mentioned the hepatic portal system as the second one we were going to see. Not lymphatic. It's a good guess. See where you're coming from. Yes, it involves capillaries, because there are definitely capillaries. All right, the other portal system that you saw was the hypothalamic hypophyseal portal system, the connection of blood vessels between your hypothalamus and your pituitary gland. So, do you guys remember what was special about that vascular arrangement? Now that we've had blood vessel lecture, we can actually talk about this a little bit more clearly. You're thinking about the pituitary in general. Yep, maybe. All right. So what's special about it is you go from a set of capillary beds. In the case of the hepatic portal system, you go from capillary beds in the small and large intestine, from those inferior and superior mesenteric veins. And that goes to a larger vein, the hepatic portal vein. And then that goes to the liver, where it goes to more capillary beds. So is it a typical arrangement to go capillary bed, large vessel capillary bed again? No. And that's what it makes it a portal system. It's a portal. It's a magic portal from your intestines to your liver. So you absorb nutrients from your intestines, and the first place you send them is your liver. And then your liver gets a chance at all of those molecules that you absorbed from your digestive tract. Now, maybe you remember we were talking about villi in the small intestine, and now they have lacteals. You guys remember the lacteals? So the purpose of those lacteals was to absorb fats. But those lacteals are not lacteals. Those villi also contained a capillary bed. Those capillary beds absorbed proteins and carbs. Proteins and carbs travel via the cardiovascular system, whereas fats travel via lacteals. Regardless of which vessel picks it up, they all go to the liver first. And then that branch of the hepatic portal vein is going to basically wash those nutrients through the cells of the liver. It's a capillary bed, so you have easy transmission and diffusion of nutrients across this capillary endothelium to these cells. So these cells are going to have all of those common cell structures that maybe you remember from AMP1 when we talked about cells, that endoplasmic reticulum. Smooth endoplasmic reticulum, rough endoplasmic reticulum. Do we remember what those did? Which one's proteins? Rough endoplasmic reticulum metabolized proteins, which means that it helps synthesize proteins from individual amino acids. And then what did the smooth do? It did help detoxify, which is another function of these cells, is to detoxify things that were picked up by the digestive system. And then what else did smooth endoplasmic reticulum do? It also metabolized. What did it metabolize? We already got proteins from the rough endoplasmic reticulum. What are our other two macromolecules of life? Carbs and lipids. So smooth endoplasmic reticulum also metabolized lipids and carbohydrates. So this is like our prototypical cell here in the liver. It's doing all those metabolic activities that we spent all that time learning about cells, learning about. Does it make sense to you guys? It's cool, right? Like we finally found that one cell that we actually learned way back in the day. It took us this long. And again, the lacteals are washing the fats over these, whereas the branch of the hepatic portal vein is washing the nutrients this way. It's also going to have an arterial supply to oxygenate that liver. That's going to carry blood from the aorta as well. And that's going to return to the hepatic veins, to the inferior vena cava, and back to the heart. So after we've metabolized it in the liver, that liver gets that first pass. It gets a crack at the metabolic activity and detoxification. Then it gets spread back in throughout the cardiovascular system, so that we can spread those nutrients to the rest of the body, because the rest of the body needs the right glucose levels, the right amount of lipids, fatty acids, and the right amount of proteins. I don't remember if I said all three of those or if I repeated myself. Sound good to you guys? Okay. These hexagons are super cool though. Like histologically, literally, you're looking at hexagons. Just like that. Each of these is called a portal triad, by the way. One branch of the hepatic portal vein, hepatic artery, and the bile duct. That's going to be a portal triad. That's going to be at the corner, every corner of the hexagon. Okay, we spent a lot of time on liver because, not surprisingly, when we get to our pathophysiology class, that liver is that's a whole chapter just for the liver. There's so much that can go wrong with it, and because it does so many things, you're going to have a lot of implications for liver failure. Okay, gallbladder. Again, the liver synthesizes the bile. The left and right hepatic ducts brought bile to the gallbladder. The gallbladder simply concentrated and stored the bile. And then when it's time, when we sense food has entered, when we sense chyme has entered that duodenum, the gallbladder will contract and release bile into the duodenum via the common bile duct. So here's the common bile duct. Note that this is the head of the pancreas over here. So the bile duct actually goes in through the pancreas and meets up with the main pancreatic duct. And they secrete their products through this hepatopancreatic sphincter, and they do say sphincter usually for this one, it's definitely not a valve, into the duodenum itself. So if you open up that duodenum, generally you can find a little hole, a little opening, a little ampulla right there. There's a lot of significance to the fact that these ducts meet up. In particular, when we talk about patho, if a stone from your gallbladder lands right about here, it's not just going to mess up your gallbladder, it's going to mess up your pancreas too. So the function of bile is that bile salts are going to help with fat. It's going to help you emulsify fat. And what emulsify means is that it's going to help fat bind to water. And that has to happen if it's going to make its way through cells, because you're mostly water. Your bloodstream's mostly water. So we've got to break down that fat into something really small, and then we've got to surround it with something that's going to move through water very easily. And that's what bile salts are going to do. It's also going to contain bilirubin. Your liver broke down some hemoglobin and needs to get rid of it. So we're going to release some of our bilirubin through bile. It's just going to be like a waste process, basically. This also means that if you have your gallbladder removed, the color of your feces is going to change. Any questions about the biliary apparatus and bile and gallbladders? If you don't have a gallbladder, the main issue is you're going to have a hard time digesting fats.
Where does it go?
Then fat is going to stay in the lumen of your intestines, and that's actually going to be problematic. It's probably going to cause things like diarrhea and some other. It's called osmotic diarrhea, where you just have too much stuff in there. Okay. I'm sure there's other problems, but that's the most literal. On to the pancreas. Almost done, guys.
Why is cholecystitis so common? Do things just get blocked up in there?
Cholecystitis is very common, you're right. A number of reasons. A lot of it is associated with cholelithiasis, not all of it. High-fat diets are pretty common in the United States, so that's going to have an impact. There's a huge genetic component to it and a huge nutritional component to it. So we will learn the pathophysiology of gallstone formation and how that contributes to inflammation of the gallbladder 90% of the time. And that we'll see the genes' names. We're not going to be responsible for learning the gene names, but some genes are overrepresented, some are underrepresented. Basically, you just have crystal formation, and it's just too concentrated.
My doctor specifically asked, because I had to have mine removed, is the fertile female and fat.
Fertile female and fat.
I only fit 2 of them. Female and fertile.
Fertile female and fat. Yeah, that's great. Good job, doctors. Always really classy. Stay classy. Okay, bile, pancreas. We've already seen the pancreas for endocrinology. We focused on those pancreatic islets of Langerhans and how they secreted insulin and glucagon. Today we're talking about the exocrine pancreas. The rest of those cells were organized into acinar glands that secrete pancreatic juices into the pancreatic duct. And again, these are retroperitoneal. Back here we have the head of the pancreas nestled ever so gently into the duodenum. And that pancreatic juice is going to contain, most significantly for us, amylase, lipases, and nucleases. What does it mean when it ends in ase? It means it's an enzyme. And some enzymes break down. All of these enzymes, yes, they all break down. Amylase. We've already seen amylase. We had salivary amylase. Do you guys remember what that broke down? Carbs. There's only 3 options here, or 4 options technically. Lipase. We've already seen a lipase before. What's it break down? Lipids, fats, fatty acids. And then nucleases. There's actually 4 macromolecules of life. Proteins, fats, carbs. And does anybody remember the other one? Nope. Organic molecules. Vitamins and minerals are inorganic molecules. Nucleic acids. So the things that make up DNA, and everything you eat was living at one point. Plant or animal, doesn't really matter. They were all alive at one point. They all have DNA, so they all have nucleic acids. So now we've discovered substances that are going to help us break down every single macromolecule of life. Amylases are going to help us break down carbohydrates. Lipases help us break down fats. Nucleases help us break down nucleic acids. And peptidases, like pepsinogen, are going to help us break down proteins. So now we have pancreatic amylase as opposed to salivary amylase here. We do have some varieties of these. This is a very specific carbohydrate that amylase breaks down. You can find other enzymes that break down other varieties of carbohydrate. We're just kind of going with the general terms right now. Pancreatic juice is also alkaline, which is really important because look at our position here. We had a bunch of hydrochloric acid up here in the stomach, and then it left right into the duodenum. And do we really want a really acidic environment right here? No, it's not really built for it. So we secrete alkaline substances to neutralize the pH. We got to get back to that 7.4-ish. I mentioned before that in your villi you have lacteals. They transport fats. So you actually have a fat globule from food that you eat, and that has to be broken down by lipases, and bile salts are going to help break that, emulsify that, and essentially liquefy that. They get smaller and smaller and smaller until they're small enough to diffuse across this endothelium. And then it's actually a process of exocytosis to get it over to this, this side of the mucosa into the lacteal. So I do want to hold you to that exocytosis for fats and into lacteals concept. Proteins and carbohydrates travel in very similar ways. That's a fatty acid, sorry. I focused on the wrong one. There we go. So this is how glucose is moving. Note that we have something called a glucose-sodium symport. What that means is we're actually going to use the gradient of sodium. There's more sodium on the lumen of the small intestine than there is inside of the villus. So that sodium wants to go into the cell. I know you can't see my icon right now, but the recording can see my icon right now, where I'm showing you how this is traveling. So sodium wants to move into the cell, and it's going to use the energy of the sodium going down its gradient to pull glucose along with it. On the other side, you're going to have a passive glucose uniport. So you're just going to have this arrangement, and it's going to be more or less the same arrangement for proteins. The monomer for a protein is known as an amino acid. So we're going to have an amino acid sodium symport for proteins as well. And it's still, again, going to be powered by the gradient of the sodium. And once it gets into that lacteal, it's going to be absorbed via a capillary bed. So again, glucose and proteins use sodium symports to cross this lumen into a capillary bed, whereas fats are emulsified with bile salts, passive diffusion, then exocytosis into a lacteal. We are going to have disease states that are specific to lacteals, for example. Destruction of lacteals like Crohn's disease, and that's mainly going to impact how fat is transported. So there is some significance to the differences in this. The requirement of having bile salts for this process means that that's part of why we're going to have a hard time digesting fats after having gallbladders removed. So there is clinical significance to this. It's not arbitrarily forcing you into learning a bunch of tiny details. I already mentioned the importance about vitamins K and B in the large intestine. Also in the small intestine, vitamins A, D, E, and K, vitamins C and some B vitamins, diffusion or active transporters, vitamin B12, absorbed via endocytosis. I do have some things highlighted in blue here. Diffuse, this word's highlighted. Diffusion or by passive or active transporters, that is highlighted. Endocytosis, that is highlighted there as well. You'll absorb a lot of electrolytes in your intestines. Again, this is significant because electrolyte imbalances are significant. I don't think that I have a lot more here for you. Don't forget that parathyroid hormone is about calcium homeostasis. One thing it does is leach calcium from bones, but it also increases absorption from the GI tract. So your GI tract will respond to parathyroid hormone. And again, a lot of water absorption happens small intestine and large intestine. Mostly by osmosis, rest absorbed in large intestine. That's about it. Water uptake is coupled with solute uptake. What that means is that we're going to absorb isotonic fluids a lot faster. Do you guys know what isotonic means? What's isotonic mean? I know you know what it means, but let's see. Equalized, right. Do you guys remember what's an isotonic saline solution? Normally, yeah. What's like the percentage? 0.09%, I think, is the right one. What that is to say is that sports drinks are correct. Sports drinks are isotonic, formulated to be isotonic. That doesn't mean they're healthy. They also have a bunch of glucose. but they did figure out that isotonic thing and speeding up absorption. Some sports drinks do not have sugar, and I would recommend those.